That's great.
I connected it to it, like the signal's strong, but it says no internet access.
No, it's working. That's your computer. It's a problem. All right, I'm gonna try to open it again. See if it works. Recent places. Cell respiration. Come on. Do it! Just do it! Alright, so while I have to click on folders here, Lab 4 Part A was about the little cubes, right? They had different sizes, alright? So I just want to make sure that we talk about all the parts of all these labs because they're going to be possibly FRQs in the test. We never know. So that was about, so somebody put it together for me. What was that lab about? Yes, Sasser. No, sorry. Yes.
Okay, so we had different agar groups,
Uh-huh.
and then they had something on the inside.
Yes, and they also have a base dissolved inside, which makes the filament turn pink. We did that as a demo right before the lab, right? You were really supposed to do it yourself, but we did this as a demo, and that was one of the things you're supposed to just say. You could tell there was a base because it turned pink. And meanwhile, if you put an acid, it drops the pH below the range that phenolphthalein changes color, and it turns black to clear, right? So that's the whole key of the lab. By the end of the lab, the cubes turn towards the clear. But does the entire cube turn clear necessarily? Now, you saw that the smaller one turned clear more, and then there was a tendency, and that one actually worked pretty well. Like, it's frustrating when labs don't work well, but that one should have worked pretty well. But what are you supposed to do with that in order to figure it out, like data, statistics? What is the right thing to do? Somebody tell me to figure out how much one is. Qualitative, you can look at the smaller one. By the way, why is the smaller one better? To volume ratio, the diffusion of acid into it is faster, right? So more of its volume. But it's also, you could also make the argument that there's less volume to change. So if you just look at it, and say, oh yeah, it's clear. You didn't actually prove that. You have to look at what to determine that that it's actually making a difference. The bigger surface area to volume formation. If you just look at it, which one changes first, that's a fallacy. All right, and a lot of you guys on your report had that fallacy. It's not because it's smaller to change first that it's proving that it's better. It also has a smaller volume to change in the first place. So that's in fact, if you really think about it. You're changing two things. You're changing the size and the surface area to volume ratio. So there's less phenolphthalein, less base to react with the acid to begin with. Is that clear to everybody? So what data do we have to look at to see if it really was better? Yes? No, not really. There's nothing. The outside doesn't do anything. Yes? Again? Yeah, but what does it mean by the rate? What do you mean by that? Because I just talked about that. It's faster to change something smaller to begin with.
Like the ratios.
What do you mean? The ratios. What percentage of the volume was changed, right? So that's important, okay? So that yes, the cube, 100% of its volume was changed in an X amount of time. You gave the same amount of time for the other cubes. What percentage of their volume was changed? So it's not about which one totally changed first. It's about, or the speed at which it changed. It's about the percentage of the volume which would change, which means you have to calculate the pink volume before and after, right? And then figure out a percentage of what was changed. So that was part A. Everybody on the same page about that? Then on part B, you set up 4 different setups. The goal of part B is for you to understand water potential. You have to calculate water potential, right? So remember, you're going to have to do this in the test It could be one. In a test, there's always 6 mathematically based questions, right? And that's one of the math things that show up in the equations, right? That the total water potential is pressure potential plus solute potential. And some of you guys really struggle with this because you didn't do the pre-lab studying that you're supposed to do, and you came here like, oh my God, I don't know what I'm doing, right? You have to prepare. But this pressure potential, what do we say about it in this lab? That we kind of ignored it because of outside and inside is the same. It's not entirely necessarily true that outside and inside are the same. There's an air pocket inside, right? So you got a little bit of air pressure inside of the thing. And then the outside, there is no air pocket. It's just inside submergent water. So there is actually a differential pressure inside of that. And that's something that we discounted that could explain some of the reasons why some of the results of you guys didn't work completely right. Another reason is that the water is supposed to be distilled that we use to create the solutions, and by the calcification that's gathering that on that sink, because Mr. Prieto doesn't close the sink every single day, you can actually figure out that there's not distilled, because if it wasn't distilled, there'll be no calcification gathering there. There's something in that water. I already complained about it, but it is what it is. So if it's not distilled water, that's going to affect things, which is why you have to have a control group. Every experiment has to have a control for you to compare with. So if this is actually something very important. Some of you guys said, oh, this doesn't work. It didn't work. But it did work. You're just too stupid to realize that it worked. And I want to say this nicely, but seriously. I want to explain what it means. Right? Like, if the control went up by 5%, even though it shouldn't be known that change. Right? And then your group, their first trial, went up by 2%, right? Well, then you're supposed to have lost water, but you didn't want water, and you say, oh, it didn't work, we gained water instead of losing it, right? Well, actually you did lose water, because you gained less than the control gained. And by that token, every group should have gained that much. Now, big flaws of that part B is that we didn't repeat it. So with no repetition, you don't have averages. Without averages, you have the idea if your data is precise, right? Each person only did one setup once, correct? Each group. So we did 5 setups, 1 was a control, but each type of setup was only done once. So you don't know if that particular time you just did something wrong, and that's why the data was like that. Are you with me? Everybody clear on that? Having said that, another thing that we don't know, without doing statistics, you don't know what that 5 and that 2 are different from each other. That's what's so important, right? Because if you don't get averages, and then from the averages you calculate a standard error of the mean. I'll give you an example, okay? So let's say you could do calculations, right? Let's say this is actually a 15% increase, and this was a 12% increase. But you do the averages, and from the averages you do the standard deviation, and from the standard deviation you calculate the standard error of the mean. This is what you should strive to do in your labs. You really need to have computational data statistics. If you don't have that, you have no idea about the effect. So all of you guys are saying these bold things. The results fail to reject the hypothesis, and you have no statistics on your lab. That's not science. You can't say that. You can say what you kind of learned from it, but you can't boldly say, I rejected my hypothesis without doing any statistics. That doesn't make any sense. I'm trying to teach you something here, right? So this 15% here. Let's say the standard error of the mean of that 15% when you calculate the standard deviation, because you have multiple trials. Otherwise, you can't even do that. You can't even have a mean, a standard deviation, and a standard error of the mean without multiple trials. Again, another reason why it's important. But let's say you get a standard deviation, and from the standard deviation, calculate the standard error of the mean, and the standard error of the mean is 5. So that means it's not 50%. It's 15% plus or minus 5%. So it could be 10%, or it could be 20%. That's what that's saying. That's how I do understand what the standard of the mean means. Right? And then you just do the standard of the mean for this one, and you get 4%. 4%. That means this could be 10, while this could be 16. So without doing actual complicated statistics like t-tests, or chi-square, or other complicated statistics that you learn in AP Stats, just that can tell you that you have no idea if these numbers are different. Right? Because at maximum, this could be a 20, and at minimum, this could be a 10. right? And this one in maxims could be a 16, and then so this is the range for that one, and then the range for this one goes from 16 to 8. But because of all this overlap here, they could be the same thing. If you fail to do this kind of analysis, you have no idea if your results may mean anything. Does that make sense? And that's something very important they can ask you to discuss on. Remember we talked about this a few days ago. When you are analyzing a lab, what are the things that they ask to analyze? And one of the things they can ask to improve, to talk about, is what things they could do to improve the data analysis. And if you didn't do that, that's something you should improve. For example, those of you who are grading labs, because of Lab 13, it requires you to do a peer review. If you did Part B, you get a Part B sent to you so you can do a review to get grades for Part C. Whether you do it or not, Part C counts against you, although you only have to do half the work, but still, right? By the way, you can always sign up for a peer review of anybody's lab report. So, for example, you do a lab, on Lab 13, it's required. But if you would like to get extra credit grades, you can always say, yeah, Ms. Lee, I would like to do a peer review for Lab 5. All right? And then you say, I'll send you a Lab 5 and do a peer review for them, and I'll grade your peer review and see how much it agrees with mine. And then you can get level points from that. Okay? It's like a voluntary homework that you're doing. It is important to do that because, remember, one of the skills you're going to have is either designing an experiment, analyzing an experiment, or talking about how to correct an experiment. A few days ago, we talked about this in this class. What are the things that we need to do when we're doing a good experiment, right? So give me something that we need to have when we're doing a good experiment. All right, so we have to have a control group. What else do we have to have? Yes? Trials. Okay, so that's steps to increase precision. That's what that's for. What else do I have? No, precision, no. Reliability. What else do I have to have? Steps to increase precision. How do I increase precision in a lab? What is precision? Is that clock more precise than a stopwatch with decimals? Why not? What's precision? It has to do, accuracy is different. Precision is about getting a number that has more sig figs, right? Getting closer to the truth. Narrow band of examination. So how do I increase precision in the lab? I have a pipette that measures something in millimeters, and another pipette that measures something in 0.1 millimeters. What are the steps I can do to increase precision? We use the best pipe. I can use centimeters or inches. Which one do I use? You see, that's steps to increase precision. You have to have the independent variable, and for that you have to tell me what it is and how it's being changed. The specific. So you can say, I'm going to control temperature. You just told me what it is. You didn't tell me what you're doing to control temperature. That's the problem with some of your designs, saying this is what I'm going to do, right? But if you don't tell me how you're going to do it, then it's a bad design. Are you with me? You have to tell me specific. This is what I'm going to do to control the temperature. And then when you're trying to do that, some people said, I'm going to do temperature control by heating up the sodium bicarbonate and then putting down the infusion. But then you're not really doing that. You're doing that just doing the infusion. During the 30 minutes the lab is actually running, it's at room temperature. You're not actually controlling the temperature at which photosynthesis is happening. You're controlling the temperature during infusion. So that was not your hypothesis, though. So your experiment has nothing to do with your hypothesis. And then I have to call you out on it. Are you with me? I'm saying. So the dependent variable. You have to say me what is the thing that's supposed to be responding. What are you interested to see? What the effect is of that on. But for that you have to tell me how to measure it. How exactly are you measuring the productivity? In this case, there was a Fulling disk going up, right? You have to have a control group. You have to have constants or things which do not change. What are the constants and how I'm going to make sure they don't change? Why? Because if I don't control the constants, they can also affect the dependent variable. And now I have multiple variables, multiple things that I'm changing. I have a group that says I'm going to control for temperature, and then in one solution I'm going to put sodium bicarbonate at something, in another solution I put, and it's like you have two variables because you're changing two things. Now you can't change two things. Now a good procedure also has steps for safety. A good procedure also has data analysis indication. What am I going to do once I get the data? And you should think about things like calculating means, standard deviation, standard error of the mean, so that you could say simple analysis like that. Understand what I'm saying? And so forth. All right? So these things. Now, in an experiment, you could be asked to analyze, to either explain, design an experiment. You could analyze to ask the question. By the way, remember, the question leads you to a hypothesis, which in turn leads you to has the variables in it, because the hypothesis is the relationship between the variables. So if my question is, does light, the type of light, the color of the light, affect photosynthesis? That suggests my independent variable needs to be the changing the color of the light, and the dependent variable has to be the rate of photosynthesis. So my hypothesis would be something like, if I expose the things to the different colors of light here, the performance will be in this ranking, as by this ranking. And then you say, standard light would be first, blue light would be next, red light would be next, green light would be next, black light would be last. Whatever you think is. Of course, this hypothesis is supposed to be based on background knowledge. Somebody sent me a hypothesis saying the green light would be better. I'm like, okay, you can have bad hypotheses which are designed to be refuted, but at that point, I mean, have you not learned any photosynthesis to say the green light is going to be the best one? They reflect the green light. It's going to just sit there, the disk. They're not going to do anything. It's like putting them in the dark. In fact, you should put them in the dark just to make sure, a control group, right? Otherwise, they don't have a control group, right? So let's see all these kinds of things. Now, you could be asked just to come up with a question that could be tested. You could be asked to come up with a hypothesis. You could be asked to justify a hypothesis based on background. You could be asked to design an entire experiment on the FRQ. You could be asked to modify the design or improve it, or identify parts where it could be better. For example, maybe the person did not control. That's a good one. That usually is a good place to attack. Lack of control. Lack of an actual comparison group. So that you don't know that your independent variable did anything because you only tested one thing. So if you did, so cigarette causes cancer because everybody that smokes gets it. That wouldn't prove it. What would prove it? That don't smoke. Are you with me? So every time you hear the news, people that smoke get cancer, does that prove is that a good statement, a scientific statement? No. A good scientific would be like, people that smoke tend to get cancer, while people that don't. You see the problem of things. This is science identifying the mistakes, lack of control. Maybe it didn't measure very precisely. Maybe you can improve the precision or the reliability. Or maybe your data analysis didn't make sense. Or maybe you didn't have any safety. So you'll be asked either to create a design or create a question or pose a hypothesis or justify the hypothesis or analyze the data. Or you could be asked just to fix the analysis or fix the hypothesis or fix the independent variable or fix something about it. But this is something they're going to ask you for sure. And this is why we're doing the labs, because it's a mechanism to practice that. And if only one person in your group is coming up with designs, that's not what it should be. All of you guys should come up with a design, right? And we barely done, for future labs, there's not going to be much designing anymore, that much, but there's still a few. Everybody should come up with a design, and then you should decide which one is the design you guys use. Understand what I'm saying? Your design is individual. Each person should come up with one, and then, because otherwise, you're not actually practicing this skill. Is everybody clear on that? All right? All of this, by the way, is in that file that I shared with you guys on General Resources. It's called How to Write FRQs. I talk about these elements of a good experiment that they're asked you to do. Everybody clear on this? All right. So it's important to take your time with that. Now, part B was about that. So you're supposed to look, and now here's another thing about part B that people fail to realize that we have to talk about as well. All right? We're still talking about lab 4. All right? If I have a setup, let's say, for example, where I have salt outside, right? And then I have, maybe inside, I have albumin, right? Now, what could happen is that when you calculate the water potential, at first, you get it one direction. But then, the salt moves, but the albumin doesn't. And then, at the end, you get a different direction if you calculate the water potential again, and by what I mean by the end. So let's say, for example, you had 1 molar and 1 molar, right? So because the concentration of the ionic constant of this is 2, the water potential here is going to be lower because the solid potential here will be higher. And since the water potential is negative solid potential, right? In the formula is negative ICRT, right? So because of that, you're going to get this number is going to be bigger. So the tendency is going to be for water to go towards the salt. But at the same time, salt's going to be moving inside while albumin just sits in there until, by diffusion rules, you're going to get the same in both sides of salt, 0.5, 0.5. Meaning at the end, you're going to have 0.5, 0.5 over here, and still 1 molar of the albumin. Which means at the end, water tends to go inside. So then your prediction should be that water is going to go inside overall, not outside overall. It will going to go outside at first, but by the end, it's going to start going inside. Does that make sense? By the way, can this system ever reach equilibrium? That's another important point. It can't. Of course not. Right? Because you can never put albumin out. So the tendency is going to continue to be to put what? Water inside. Of course, at some point, the pressure becomes so high that the pressure potential, the other one, very good point. Right? When the pressure potential becomes too high on the inside, then it does reach equilibrium. But if you look at only solid potential, it doesn't. But you see why we did not... So everybody who had to explain bad results should have invoked pressure potential as a possibility. If your bag got to the point of getting taut, are you with me, full, that it can no longer take water, then it stops the process of what it's supposed to do. So that's something else that Lab B is supposed to realize. Then on Lab Part C, it was just about seeing the cells deflated, inflated, and that was really cool. Those of you who didn't do it, make sure you do it. It's super easy. Grab a little leaf from the thing, put it on the microscope, put some salt on it. So, who did it? All right? Wasn't it cool to see the leaves losing thing? By the way, I do have a video on the cell membranes, videos in my tutorials that shows you that, and it was part of the pre-lab to look at the video anyways. So even those of you who didn't do it, you saw what happens. The salt comes in, and all the cells kind of deflate. The shape still the same of the cell, but then you can see the plasma membrane squeezed in, all the chloroplasts kind of squeezed in there, right? And then as soon as you put fresh water, it goes backwards, and it fills up the thing again. It's a really cool thing to do if you haven't seen it. Then part D, right, was about testing a single variable that could affect the fusion, temperature, the concentration rate, or anything else you wanted to. And then it's a design that you made on your own. So part B was all about this, actually doing an experiment. Now, if you do an experiment and you don't discuss the data of it, you're not doing a great job. Not doing a great job. Okay, so before I continue into Lab 13, for the sake of our course, I'll put that on the pending list, and let's go ahead for 4.B.2. So 4.B.2, I still owe you that video, the animation from cell respiration, but forgot to click on this. It's going to open sometime soon. So those of you who have the essential knowledge, please open in 4.B.2. I'm going to project it, but it's not going to be very big. So 4.B.2 is about the mechanisms that life uses to conserve energy in an effort to use less of it. All right? We talked about this briefly doing 2A2 when we mentioned that endothermy, that there are mechanisms or behaviors, either mechanisms, internal mechanisms, physiology, or behaviors that the life engages with in order to save energy. So for example, physiological mechanisms with plants of flowering at the right time, to have the offspring at the right time. Physiological mechanisms in animals to go in heat at the right time, to have children at the right time, to reproduce at the spring rather than the winter. But also the courting behaviors which engage during the right time for that to take place. We also talked about endothermy versus ectothermy and the behavior of going away or to the light. By the way, we are endotherms, which means we regulate our body temperature. But do we still kind of act like ectotherms? Think about it. Do you avoid the heat sometimes? Of course you do. We live in Florida. Are we doing something to avoid the heat? Yes, we go inside to the AC. Because we could be having this awesome conversation outside in the field. But we'd rather be in a controlled temperature environment so that our bodies don't work so hard. So you go outside in the damp, heat, warm Florida, you're like, oh my God, can I go back inside already? So aren't you like a little lizard after all? So I like to talk about this because it's not as if we don't do what they do. We do more than they do. We also have a few, so we are evolved. We were like them, where we gained something. We had a derived care that they don't have, which is being able to regulate or internal. Does that make sense, people? Right? Also, remember to talk about this. Respiratory rate matters among endotherms. The ectotherm respiratory rate matches the rate of the environment, correct? Which is why during the winter, if you go out at 9 or 6 o'clock right now, you don't need bug spray. Because what are the bugs doing right now? It's too cold for them. They can't move around. They can't fly very well. Why can't they fly very well? Because their respiratory rate depends on temperature. So they can't maintain flight with those kinds of temperatures. So they don't bother you because they're hibernating. Literally, all the birds are hibernating. Meanwhile, us, because we're regular temperature, we can do it. But we can stay through winter. But even large mammals hibernate or estivate during the summer hot months. That's called the opposite of hibernation, estivation, which is when you avoid the heat months and you sleep through the summer in the desert. So a lot of mammals do that. They live in the desert. So we talked about some strategies already to conserve energy. This is more about a different angle on this. It's interactions between cooperative systems, either at the level of population or community, right, or at the organism level, in order to maintain or save energy. So organisms have components which perform specific functions, right? Now this is another thing that used to be in the older AP Biology course. It used to be like an overarching theme, the idea of emergent properties. So before we talk about that, let me plug in something that's no longer in EK, but it's actually kind of, I like it. It's important. It's an interesting phenomenon about life. So let's think about this. At what level does life emerge? Level of organization. Can molecules be called alive? No. Prions are self-replicating RNA or DNA molecules that cause disease, or proteins even. There's some viroids or some stuff like that that's not even alive, but it has self-replicating functions. But we don't call it alive because what do we need to call it alive? We have a characteristic of life. And those things don't have one of them, which is what? They don't actually try to maintain homeostasis. They don't actually are made up of what? Cells. So at what level do you see life starts? At the level of organization. A cell. At what level does communication start? Do viruses talk to each other or understand other things? Information exchange. At what level does that happen, do you think? Ever stop to think about that? Viruses can recognize their target cells, can't they? So somehow their protein membrane is able to detect the surface proteins of the host cells in order to infect the right cell, the cell that they work on. That's a rudimentary type of recognition, not necessarily communication, but at least recognition, right? And establishing that that's my target, right? But you wouldn't argue for true communication until you talk about sex and cells needing to talk to each other to reproduce. So it's a cellular level that happens as well, right? It's a basic communication. Or a cell telling another cell of your body, like the pancreas, producing insulin to tell other cells on the liver to put things into glycogen because there's too much sugar in the blood. That's communication across long distances, right? Right? Organelles within the cell that they really talk to each other. They talk to each other, the organelles, somehow. What about the ribosome that's free, but attaching itself to this raw VR if he's bound to send something to the outside? That requires a level of kind of management and organization. So you see, emergent properties, if you think about a property, where does it emerge? Talk about intelligence. At what level does intelligence emerge? That's a big question. Is a dog intelligent? Is a bug intelligent? It depends. Does a bug make decisions? At what level does decisions arise? That's very interesting. Was the Australopithecus australianus, one of our ancestors, was it intelligent? When did that spark of intelligence light up? See, but that's an interesting question. Adam and Eve. Does that make sense? That we all come, is there a point in the line of evolution that say, oh, that's the Homo sapiens? Or is it so gradual that you can't tell where the new species is a new species from its ancestor? It's hard. These are all interesting questions. Now here's a more interesting, deeper question if you ever thought about it. Consciousness, self-awareness, knowing you're alive. At what point does that thing? And is that particularly... Now, we all think about that as a human brain function, correct? But because nobody thinks about another animal having that self-awareness. I know I'm alive. I know I'm in pain. There are some higher monkeys that understand pain, understand sorrow, understand loss. Okay? But maybe not... So is consciousness perhaps not like evolution of the Homo sapiens species, and perhaps also a what? Gradual progression, such that it's not a switch, right? At which point does if you believe in the soul, I'm not saying it's not a biological question, but it's just an emergent property. I'm trying to practice that in your mind. At what point does the living soul emerge, if you believe in that kind of stuff? What is alive that has a soul? See, emergent properties. Here's an interesting question that Neil deGrasse poses a lot. He likes this thing. I watched 6 or 7 videos by him, and he's always mentioned this one, so I like this one. We're looking for intelligent life, right? Outside there, you're just looking for life, period. But intelligent life would be nice, wouldn't it? To find other life outside of the world that was intelligent. Now, just think about this. The biggest, or the most ancient radio signal sent by humans, right, was something from the early radio days, and that has not even traveled, 70 light years yet? And yes, there is some good candidates within 7 light years for life, but not so much necessarily for intelligent life. But if there is a intelligent life within 70 light years, they just got our signal. Even if they were replying to it, if they could understand what the hell they're receiving it, right? We would take another something light years to receive a message back, assuming they're transmitting at light speed, and not some other type of light speed breaking technology that we haven't even conceived of yet, because as we understand, you can't break that light speed, right? But imagine that it's somehow possible to warp space and time, or do wormholes, or even travel faster than the speed of light. And this is technology that we can't even fathom yet, right? Imagine that this is possible. A race that intelligent to produce something like that, think about it, that intelligent. So, do we talk to monkeys about how do we feel? Do we get to monkeys and say, I want to go to the movies today. Would you like to come with me? Do we ask monkeys out? Do we hang out with monkeys? We have meals with them? Do we have existential conversations with monkeys? Right? So imagine a race that powerful, that intelligent. We are only 1% different in DNA from a chimp. Now, this is an interesting thing. That 1% hides a lot of things because, and this is a point that Richard Dawkins, another important, famous evolutionary biologist, if you like, you can listen to him. He's really awesome. But he would say, it's not the difference in our DNA between the chimps. It's not just the 1%. That doesn't mean we have 1% more than they do. First of all, we have 1% different than they have. But we are different from them more than just because of what we have that they don't have, or that we have that's different from what they have. But because of the things we have that are the same as them, used in different ways. Our DNA activates in different sequences. We'll learn about this soon. So, being who you are is not so much just what you have, but it's also the order and sequence and timing of when the genes turn on. For example, we have the genes to make our chin long like they are. The same. But they never turn on, so our chins are like this. They're not expressed. So that means, in that sense, we are different from them, not because we don't have the gene, but because our gene never turns on. So you can create a lot of difference with 1% difference, because it's not just about what you don't have, but about how you use what you have. Does that make sense, people? Right? Having said that, imagine 1% more different in the same direction as the direction that we are evolved compared to them. Would we understand them? Would they want to talk to us and have existential conversations? And if there's an alien passing by, would it be not we worms looking up and seeing a human walking by and wondering what the hell that is? would we even recognize that what the human is doing is intelligent? Think about it. Does the worm contemplate human intelligence? Does it even understand the emergent property of consciousness? So, can we even imagine what emergent property will come next on evolution? So, maybe what we call consciousness, what we call intelligence, is something that their babies can do. Maybe they look at Stephen Hawking, oh, look how cute. He can do astrophysics in his head like little Timmy over there. That's a joke that Neil deGrasse Tyson uses. But it's true. Right? But having said that, biological systems work together because each thing, I'll give an example. This is what is this? An hourglass. Is it made of one thing or is it a system? It depends. I can call it an object, an hourglass. But I can also call it a system made of many parts. Are you with me? Now, what does it do? It's an awesome emergent property that this thing has. It can tell time passing by, right? Another stupid concept made up by humans. Think about it. 3 years ago, you were somewhere. A four-dimensional kind of thinking makes you realize that you're still there. That hasn't changed. That happened, and it happens still. It's happening now. All right? If you think about, have you seen Interstellar? The idea at the end of the movie of the Tesseract is that if you could travel in time as you travel through space, every moment in time is just as accessible as every space. But we are confined in that dimension of time. We cannot travel in that dimension, right? Or can we? But anyways, this thing measures the forward motion of time, I suppose. Can you do it without the bottom here? If I took this bottom out, would it work? Yes, it would still work. It just couldn't work backwards. Very good. Right? But, so the real emergent property is as a reversible, reusable time measurement device. Correct? But if I take this out, I lose that emergent property. Correct? So, why does life collaborate? Because if I took the bottom out, I lost the function. So you see what's cool about evolution? You don't have to create an eye from scratch. You create an eye by adding a function to a previous existing function by adding another structure on top of it. There's a video on the evolutionary theory video lecture, videos and tutorials that talks about the evolution of the eye. It walks you step by step and how this amazing machine could have evolved without the help of a designer. And by the way, those of you who see problems with that, I'll tell you this much. Any decent second year MIT engineer can make a better one. Eye. Yes, or eye sucks. So if this is designed, good job. See what I'm saying? It's not designed, it's flawed, mistaken, constrained by the history of evolution that made us, our ancestors had eyes that were not good, and because of that our eyes will never be good as we could have good eyes. Because we are limited by the type of machine that we came from. Does that make sense? So if that's an argument against design, that's the one right there. Because any engineer could think of something better than this. Right? That, how many of you need eye prescription? So there you go. Flawed. Flawed. Now, having said that, why do we compartmentalize? Why do we compartmentalize in life? We learned about this already. Kevin, why do we compartmentalize? More efficiently doing jobs. But why put compartments together then? So that's why compartmentalize. But why have different compartments all near each other together? So that you can do what? An assembly line. Does that make sense? Okay, good. You guys said that. Okay, so organisms have compartments that perform a subset of functions. And then together, they can perform a higher function which is related to energy and matter as a whole. So this is examples of at the cell level, the plasma membrane, the cytoplasm, and for eukaryotes, organelles, all contribute to the overall specialization and functioning of the cell. So Jenny, what do you think is the job of a cell? If you have to say what the cell's mission is, what would that be? What are cells for? What are they doing? I like to replicate, yes, to continue, to pass on. But in order to do that, there are factories that make what? What is the whole point of the cell for? No, they're not making that energy. What are they for? They use energy. What is their job? Yes, Justin. That's what they do. They're protein factories. So it means to make proteins, do you need a cell? Careful. Is it an emergent properties of cells? Make proteins. Not really, because otherwise, remember, I'll talk about this, origin of life may have come, proteins may have come first. Correct? However, the cell allows you to make in fact, today, you want to make some insulin? We use a cell for that. We don't synthesize using a machine. We put it inside a bacteria, and bacteria does it for us. Because as of today, cells are still the best protein-making machines there are. That's pretty cool. Right? Probably not forever. One day we can synthesize proteins quickly. Right? They're no longer the best DNA copy machines there are. Right? There are actually machines to copy DNA and sequence it faster than cells can do it. All right? So, PCR, for example, learn about that at the end of the year. Beats the cells a lot. I still use this, though. PCR uses an enzyme that comes from cells, but it's not done in a cell environment. So cells are not the best DNA synthesization machine, but there are machines that make proteins better than we can do. The ribosome is an excellent machine. And until we can figure out a better machine to do that, it's going to still be like that. But the point is, as a whole, to make a protein, every part of the cell collaborates as a small function to the whole. What is the lysosome's function in that picture? What's the lysosome there for? Yes? Remember, I'm talking about making proteins. So how does a lysosome contribute to making proteins? Yeah, but I'm talking about saying how does it make a protein? Because everything has to relate to that, yes? It breaks up the bad ones, which gives me back the amino acids that I need to make new ones. Very good. How does the vacuole help in the making of proteins? Yes, yes? No, vacuoles don't store energy. Maybe the glucose that you need to make the energy. Very good. All right? They can store ready-made proteins for the future that I need them. Right? How? They control the osmotic pressure that I need to have a functioning cell in order to do what I need to do. How does the Golgi apparatus collaborate in the protein synthesis process? Come on, people. You're taking a test on this soon. How does the Golgi apparatus contribute to the big picture of making proteins? Yes? They modify them. They package them. Put them where they're supposed to go. Send them to the right place. So each organelle is part of the overall picture. With that same mentality, now let's go to the multicellular organism level and think about this. Specialized organs will contribute to the exchange of gases. All right? So this is what I'm going to show. It's really difficult to cover more than one EK in a day. I don't think it's going to be possible. But let's try. This is gas exchange evolution. So if you just do this, by the way, this is on the... now there's on the videos tutorial there is videos on this, but there's also I have on cable biology on the chapter about this. So the evolution of the respiratory system. Sometimes I wish you guys had the books on you with the class, but it's okay. Nothing that Google can't fix for us. I'm just trying to find a good one. Actually, you know what I'm going to do? Open the actual textbook. But you said it was chapter 40, didn't you? We'll see which one opens first. My resources or the textbook over here. All right, take a break, stretch up. Get ready for more in a second. I know, but this idea of emergent properties is kind of... Have you guys seen the movie with the blue people, Avatar? Yes. right. You know, now, do you guys realize that the planet is like alive and all connected, it's intelligent and all that? And that everything, every single intelligence of the planet is only a part of a larger system, which is, it's okay. Now, what's interesting is that, do a metaphor. Yeah, it is a metaphor, but not for the cell, I think, for the brain. Think about our neurons. Is a neuron intelligent? And one neuron, is it intelligent? Is 2 neurons intelligent? Is 1,000,000 neurons intelligent? Actually, not yet. A little worm will have 1,000,000 of them. It's like, you know. But what about, so see, at what point does the network of interconnected cells get that? Are you with me? And at what point computers will do it? Is the internet already there? 1,000,000 connected processing units? Maybe the internet is sanctioned? But this is actually an interesting question. Does the internet, if it is a thing, have emergent properties that emerge from it, which are bigger than each of its constituent parts? Based on what we just talked about, it must. Correct? So there are studies done on the way the internet works and the way that things wave through the internet sometimes, that they actually realize that in some ways there are things that happen that are bigger than any one of... Who took a psych already? Okay, so you guys can understand what I'm talking about. It's this idea of, is the parts, is the whole more than just the sum of the parts? And it usually is. When you add the parts together, you get something new. Are you with me what I'm trying to say? Now, the movie was talking about the fact that the biosphere of that planet had a function, an emergent property that was superseded that of each individual. It was bigger than the whole. Each individual was just like a neuron in a bigger brain that the planet was. They were all what... We don't need the planet of Avatar for that to be true. There are things that the biosphere does which mirrors what our brains does, in the sense of like it responds. Not necessarily intelligently, but while you're looking to the same emergent property that emerges at the brain level, at the ecosystem level. If anything, it will be a different, more encompassing than intelligence. So in other words, I would argue that intelligence is inferior to whatever emergent property exists at the biosphere level. We just don't have a name for it, but the way the Earth acts, so to speak, is an emergent property that is built of all its parts put together. Does that make sense? You're blocking the view of people right behind you. Sit to the side if you need to stand up. All right? So chapter 40 talks about animal form and function. And in the very first chapter, all right, we'll get into the basic principles of animal form and function. And then we'll talk about the progression of some of these things. If you have a simple cell, okay, well, by the way, we're just doing a survey of pictures here, okay, really quick. But if you have a simple cell, all you need to exchange matter and energy, right, is across the membrane, right, which is one of the things in the test. Diffusion across the membrane is all you really need. Even if you have 3 sets of cells, say, for example, an epithelial cell, an internal layer of cells, and another layer in between, right, it's still good enough. You can exchange liquid. For example, if you look at a coral or a sponge, they have a cavity that the water goes through inside, and then the water seeps out of the cavity to the cells. But by the time you get to 6 or 7 or 8 or 9 or 10 layers of cells, the cells at the bottom of those layers, I'll give you this as an example. It rains on top of you, okay? Raindrops falling on my head. Do you think 12 layers down, your cells get wet? This is a very interesting question. Maybe never stop to think about it. Right? It rains, it rains, it rains, it rains on you. Now, by diffusion rules, if there's soaked water over here, say you are in the water, right? And you're soaked there. Do you think that that water slowly seeps through the inside of the skin by diffusion? Shouldn't it? But it takes so long that you could be in the water all day and the 12th layer of your skin will still not get that water. Now, the upper layer certainly responds to the osmolarity because you know it wrinkling and stuff like that. Although some data actually suggests that that wrinkling has nothing to do with your cells losing water. It's actually an evolutionary thing to make us be able to grasp better underwater. It's a natural made glove. Right? And in fact, if you could even argue that when we sweat, it makes us slippery, but you wouldn't sweat a lot, you actually gain grip because the same thing kind of happens. Right? So it's interesting. But either way, there's a problem in animals. Okay, now this doesn't exist. It's not a real thing. Okay? But I'm showing you that this is the whole point of the first part of 4.B.2. That anything big enough to have a problem with diffusion, where the things can't just diffuse, this cell here would starve. It would get no sugar, no water, no salts, no nutrients. Because by the time the cells get, it's like having thylakoids, right? They stack to get the light, don't they? But why not stack a million of them? At some point, you're not getting any more light out of the, you see, all you have is green left. Does that make sense? So it doesn't become, so think about it, because life kept stacking them, right? But at some point, it stopped stacking them, because stacking another one would no longer provide what? That's always the reason. Always the reason. Why didn't life take a step further? So are we getting more intelligent? Still, our race? It seems to be the case, right? It seems to be a pattern. I don't know about now, but I'm saying, historically speaking, for the last millions of years, have we been getting more intelligent? Yes. Why has that pattern continued? Because it's still advantageous. When we hit a wall, that intelligence is no longer advantageous. Will it continue? No. I think we're already there. We're destroying ourselves. But that's a different story. All right? We got too smart for our own sake, I think. But either way, maybe not. There's hope, right? There's hope, depending on the way you look at it. Now, by the same token, there's a point where stacking these cells, diffusion won't work anymore. You won't get enough nutrients to the center. Does that make sense? And certainly not the same amount. Which would start, we're talking about how systems work. This means that the inside of the system will work worse than the outside of the system. So the cells on the inside would be not as productive as the cells on the outside. Now think about it. Does that make sense for life? Where's our most important organs? Outside or inside? Inside. Protected. Shelter from the environment. So if you get stabbed on your arm, are you going to die? Maybe if you cut the big artery, but if not, probably not. If you get stabbed just like across your chest, are you going to die? A little graze. Not really. Right? So it's hard to be impaled. How many people have lived 15 years without being impaled? Congratulations. All right? It's a difficult accident to get into. It really is. How many situations do you get into every day that can make you impale yourself? And as I say this, the roof collapses and I get impaled. All right, but it would be funny. But that's okay. My clone will pick it up. Now, do you guys understand why diffusion is no longer feasible here? Are you guys with me? Okay. So what do we do instead? What's in between every cell in your body? So this is important too. It's part of this topic. It's at the end of the cytology chapter. So if I have a cell here and another cell, is there something in between the cells? Well, some cells have gap junctions that actually literally tie them to each other. But most cells have a network of proteins and fibers called the extracellular matrix and fluid between them. Interstitial fluid is what they call it here. The fluid in between. So cells solve the diffusion problem by making sure they're all embedded in water just as they used to be when they evolved millions and millions of years ago. Every single cell of you is still living the same way they lived back in those primordial oceans. In a soup. Right? That's the way life works. That's why 90% of the biomass of plants is still water. Because cells need to be soupy. Does that make sense? But how do you provide the inner soup with the nutrients that are in the outside when you are huge like that? The answer is already on the board. How does that get provided? By a network of what? Network of what? Blood vessels. You have a river within. Right? A river within that carries the nutrients throughout your body. Anything that's large enough needed to evolve this circulatory system. of some sort. Does that make sense? But then you also have a gas exchange problem. You have to have an organ to capture oxygen and deliver it to the conveyor belt so that everybody gets the same oxygenation. You also have to absorb nutrients. That's the digestive system, right? And of course, the conveyor belt has to pass there through as well in order to pick up nutrients to deliver to the rest of the body. You also have to get rid of toxins. So there's another system that's not shown here. Well, it's shown here, an extracellular system to pick up the waste and take it out of the body. Metabolic waste like urea, like hydrogen peroxide, things to get rid of your body. Too many salts. To maintain homeostasis, you need to get matter carrying energy in, matter carrying oxygenation in order for you to burn that matter with, right, in, the waste out. By the way, the carbon dioxide does not come from oxygen, it comes from the sugar that's burning, correct? And then the actual water, the oxygen, is coming from water that's actually consumed through the digestive system, right? Water is also excreted to carry the waste with it. That water usually comes from byproduct of digestion, which does hydrolysis, right? So think about it. So or every time you build something, you do dehydration synthesis, you take water out. That produces some water. So there's water in and water out in your body. But the point is, these systems are working together. You see that? To save energy. To save energy, these systems are working together. All right? Let's look quickly at the evolution of some of these systems. By the way, the lining of small intestines are full of little villi. Why is there so little villi instead of one continuous wall? The alveoli are tiny little balloons. Why one tiny little lots of tiny little balloons instead of one large one? And the capillaries are the same thing. So there's lots of little vessels. So they are also talking about that essential knowledge there. Okay. By the way, this chapter is already due for you to read. All right. So continuing this, each of these systems performs a function. That function emerges from the properties of the system's constituent parts. But the more important part of 2AB, 4AB, is that these functions work together to perform the larger function of saving energy and consuming matter and matter energy exchange throughout the system. Which brings me, by the way, to Lab 10. On Lab 10, you were supposed to diagram the matter and energy flow in and out of the plant. It's too late, it's already due, but just so you know, how does energy, talk about energy first, okay? How does energy enter the plant? Energy. What process puts energy there? Yes, so what's this arrow? Sun is over here. And what's the arrow? Photosynthesis. All right, so now how is the energy inside? Where is it? Inside. Where does it go to? How, in what form is the energy now? Where does it go? Where does it get put? Where in the sugar? And it's not just sugar. All the biochemical molecules are carrying that energy now. It's the bonds, the chemical bonds, okay? So it becomes chemical energy, yes? Potential energy now. Chemical energy. Now, how does the energy go out of the plant? All right, so you can talk about, there's another process that's basic here. There's heat, right? It goes to heat and a process of heat release. Where do plants release most of their heat? Come on, people. Where do plants release most of the heat? Just did a lab on it. Where? Stomach? No. Through the water. Because as the water evaporates, it takes the heat with it. So it's the leaves are plants' radiators. Remember that. All right? What else? Is there another way that heat that works? But plants also do work. The plants move around, they grow, right? And then you're going to use this to do work. All right? So heat release in these processes are powered by cellular respiration. All right. Okay. What about matter? Now, I'm not going to do the matter one, but this is what you need to do for it. How does matter come in? Now, inside, of course, that's going to go to different things. Proteins. We did a whole EK about this. The first EK of this topic was about this. Nitrates come in, all right? Nitrites come in. And that goes into protein and DNA because those are the things that have nitrogen in them. Amino groups and the nitrogenous bases. All right? We have carbon dioxide that comes in. By the way, this one comes through the roots, root absorption, right? So that's the process, root absorption. How about CO2? How does it come in? It comes through the stoma, basically, right? It goes through the stoma through photosynthesis, carbon fixation, correct? And then where does it go? To all carbon molecules, all macromolecules, because all of them have carbon in it, correct? All right? What else comes into the plant? That's crucial. We have phosphates. And where do the phosphates end up? They end up in ATP, because ATP has phosphate on it. Not just ATP, actually. Any nucleoside triphosphate. ATP, GTP, CTP, all of the other, UTP, all of them. They also go to the phospholipids. They also go to DNA, nucleic acids, right? So continuing this. What else goes into plants important? Sulfates. Sulfates have to go into plants. Why do plants need sulfur? To make proteins. Proteins all take sulfur in them. All right? How about water? Yes, somebody said water. Now, this is interesting about water. 99% of the water that comes in, this hydrogen here gets dumped into all the macromolecules. The oxygen passes through the plant and leaves through the leaves. All right? It leaves to the stoma. Right? Stoma. That's a product of cell respiration. Interestingly, though, oxygen also comes in. Right? Doesn't oxygen also comes in? Because they have to do cell respiration as well. Which means carbon dioxide also comes out. Does that make sense? Now, can the plants reuse some of their own carbon dioxide? Perhaps. The ones they produce, they can get some of its own carbon dioxide back, surely. But they actually release also carbon dioxide. They also release water through their pores. Right? Actual water, 90% of the water that comes in, 99 actually, does not get broken down through the photosynthesis. It actually just leaves to help cool off the plant, to keep the thing going. None of you did a very good matter flow diagram that shows all these things. And by the way, I'm excluding things like micronutrients, which are also important to make vitamins and things like that, right? And other things which help all this happen. This is something that you should practice. It could be an FRQ as well. We're going to return to FRQs after we finish the battery of labs in January, and things like this could be your FRQs, right? Except that, unlike the FRQs in the first quarter, the FRQs which are coming will be comprehensive. They'll be about everything, right? So they'll throw things at you in preparation for us to study for the test. Okay, so now, this is the digestive system of a higher level mammal, and they're going to break down the different types of tissue. So it's a little review of anatomy for those of you who had it, but it's not the point. The point is actually talking about how is this evolving. So let's go. This is actually important, the types of signals. There's local communication versus long-distance communication, but we'll talk more about that later. This is such a fundamental chapter. If you have not read it, it's going to be on the test for sure. It's one of those things that professors don't talk about in class, but they're going to hit you with in the test. And what this chapter is about, seriously guys, I plug in things from this chapter throughout the year, right? But I never tackled this chapter at once. You know what I'm saying? So it's important that you read it. It's going to be on the multiple choice. It's like the animal behavior chapter was. I didn't cover it yet directly, but I'll plug it in throughout the year. And so you need to be able to read this. It talks about how animals maintain homeostasis. What are the processes that animals do to do that? And what are the forms that allow them to perform that function? What structures allow animals to do what they do? For example, this particular chapter here is getting into how do we maintain our temperature regulation? What if it's too cold? What if it's too hot? What is that natural thermostat? How does that work? What is the biochemistry behind it? What is the physiology behind it? All right? So it's an important chapter, but what I'm getting to right now is just the evolution of the systems. So let me see if I can get to that. We already talked about that, see? The ectodermy versus endodermy. So we definitely hit some of these things. This is actually an interesting one. In every single mammal, in every single bird, part of our regulation mechanism is to have something called a countercurrent system. Now you see that the blood vessels go down, and then they come back up from the extremities. Right? Now notice what the diagram is saying. As it goes down, the blood is warm. Have you ever felt your extremities to be the coldest parts of you? And that's the ones that go, if you go into ice, they fall off because you get the what do you call them?
Frostbite.
Frostbite. They're the most likely to actually become like that because, they're further from the parts of your body which are the warmest. But notice what actually helps them keep warm. If you keep pumping hot blood down, right, the left side is always harder than the left side. Does that make sense? So it means by diffusion, even as the blood is going up, it's warming up. Does that make sense? So I'm already warming the blood up with by the heat that I'm supposed to be losing to the environment, right? By pairing up the hot going down with the cold coming back up, that warms myself back up. By the way, some organisms have extremities of different color. Have you seen little rabbits and dogs that have their paws are different color from their hands? Right? That's usually epigenetic regulation. What that means, in translated English, is that those genes turn off or turn on depending on temperature. And since those are the coldest or parts of your body, that probably means that either genes turned on or turned off. Right? And so they don't turn on in those areas. That's why they have different colors on those paws. Which means if you shave the hair off of a bunny of that type and put a little warm thing and leave it there while the hair is growing back, what should happen? It should grow back the same color as the rest of his body, right? Until, of course, you take the thing off and then the hair will return to... You see, it's interesting that works. We don't actually do that, but that's kind of like that. So, digestive system... just looking for specific pictures. There's a picture on each chapter that talks about the progression of the evolution of these systems. Yes. I know. But I don't want to do both pages because I actually want to show you this. So, at its simplest, a digestive tract is just one continuous path. Do you see that? Of course, notice that the early stages of specialization of evolutionary process, you got some compartmentalization with different. So you have the crop, the esophagus, the pharynx, and the gizzard. The gizzard is just a spot. It's an early stomach. Does that make sense? Where food can get macerated, divided, broken down, right? And so, and the large intestine to absorb, which hasn't changed much. Of course, ours has gotten larger. By the time you get to a bug, those same kinds of specialized compartments have become even more differentiated, all right? And they actually have these sides, gastrococci. They have larger foreguts, midguts, but it's a layer of additional compartmentalization and specialization to... So you see these gastrococci are little folds which became specialized, full of enzymes to produce material to provide. We have those too. It's called a pancreas. It's called a gallbladder, right? But ours are much more developed. By the time you get to something, the simplest ones is, like I said, just a tract that absorbs things. By the time you get to the worm, it's more complicated than that one. The esophagus of a bird or a mammal is even more complicated with an actual stomach. Large, folded in large intestines and stuff like that, right? So if you get to a mammal, it's even more complicated than that, because we have folds and inner folds, and then folds within the folds, the little villi, and then we have crops and gizzards. We have a liver, we have gallbladder, pancreas, and the stomach with its own, all of it to provide a more specialized subdivided functions. So evolution is just progressively adding to it, modifying what was there before. Does that make sense? And each new structure allows to perform a new function. It actually shows you a diagram that shows you that at the end of the day, it is just one continuous tract with specialized side compartments to provide an additional structure. Does that make sense? Just like that was in the bugs, except a little bit better. All right? Well, not better. Different. More changed. You know? Along the same lines, circulation and gas exchange mechanisms have evolved progressively. And the cool thing about life is that you can see these steps throughout the history of life. So you see how the circulatory system of these worms is just like a network of blood vessels that just kind of, not blood vessels even, it's just, now this is the respiratory one. They have an open circulatory system. The heart pumps. By the way, they don't breathe through this. That's why it can bear to be open. They don't need to circulate gas exchange. The limiting factor for death, the most important thing here, waste, you can probably live on a wasteland for a little while. In fact, you can have your liver shut down, your kidneys shut down, and live for a few days. Right? you can have your stomach shut down and live for like a month, right? So digestive system is less important, perhaps crucial, because we have stored sugars in fat. And the waste system can also be less important, although you don't want to be doing dialysis every day, and you don't want to have to need a liver transplant if you're going to, a liver failure kind of situation. And when one organ fails, the others won't start to fail. It's like a cascade reaction. But respiratory system, you die in what? Minutes. But since their respiratory system is not tied to the circulatory system, they can bear to have an open, inefficient circulatory system, which basically pumps sugar from the intestines into the rest of the body, right? And they get bathed in this thing, and then little by little the liquid seeps back into the heart, pump it again. But that made them become small. It can be large like this, right? Because it's an inefficient way to deliver nutrients to the rest of the body. By the time you get to simple worms, they actually have closed circulatory systems, which actually have these heart that looks like a machine, and they have actually many hearts throughout the body, each one of them pumping around to allow this distribution of those things. They're segmented, so they have these like repeated segments of hearts. By the time you get to something like, by the way, bugs breathe through tubes on their skin. And the tubes are like a network of kind of like AC systems are in buildings, so the air gets through it like that. So by the time you get to fish, look it up. So this is evolution. But remember, guys, I'm showing you each system separately, but remember that the point of 4.B is also seeing how all these systems collaborate to save energy for the organism as a whole. Correct? All right. So then, so you have two. By the time you get to a fish, they have actual chambers. Why? And with the actual valve. Because if you had a heart that just pumps like that, some of the blood pumps backwards. And that's inefficient. So anything that would add a system to prevent backwards flow would become an advantage. And so eventually, gradually, that actually evolved. A little pump, one chamber receives the blood, pumps into the main chamber, which pumps to the rest of the circle, but with a valve that prevents it from having blood flow. That's a cooler system, except with this system, the blood has to go through the gills to be oxygenated, and then to the rest of the body, all in one pump. That, again, limits the size of the organism. And there are some large fish, but they're very lethargic compared to the movement of higher mammals that can do. On frogs, you have 3 chambers, right? So now you have received the blood, you receive the pump, and now this pump sends it to 2 different places at once. One towards the lung and then back to the heart, right? So it's kind of like our heart except an in-between, right? See, this is the cool thing about life, evolution. We have a picture because we see in the animal kingdom the progression. So even if you weren't there to see it, we can see the progression. So, of course, a lot have to happen between here and here, but there are, in the animal kingdom, lots of gradual examples of how you get from here to here, right? So, organisms that have intermediary performance between the fish and this frog that we're talking about. But either way, the disadvantage here, of course, is that the blood that's oxygenated is mixing with deoxygenated blood. Some of it, which is already oxygenated, is going to the depth to get oxygenated again, right? And meanwhile, some that's not oxygenated is going to the body. So it's a waste, again, of pumping power. So, of course, reptiles have almost a complete system, except reptiles have an extra loop that also becomes a little disadvantageous. We just have an efficient 2 hearts in 1 system that actually pumps 2 separate circulations and allows us to be faster, bigger, and better control. Crucial, animals and birds do the same thing, crucial for temperature regulation. And so that's what allows us actually, a lot of scientists think that that's why temperature regulation evolved in birds and in mammals, is because both of us evolved that heart first. The next step of physiology to regulate temperature is a subset of that. Reptiles have an intermediate performance heart that does not allow them to do as well as we do. What I was trying to show you with these systems, that's the loop of the reptile heart. They have no, that's actually the human heart. That's very complicated. Anyway, circulation. Now, the last thing I want to talk about is that even if you look at where is the endocrine system now? Excretion systems. All right, so here. So these are the excretion systems. Look at progression again. Come on. Or he's already in the advanced. On fish, a lot of the excretion happens across the gills. So they repurpose the gills for actual excretion. On large, simple worms, it's just by diffusion across their skin. But by the time you get a larger worm, you need to have a specialized system to get rid of it. And so larger worms actually have these things called flame cells, right? Flame cells, which are little bulbs that collect the toxins and excrete it through the skin. By the way, you take 3 minutes away from me in the beginning, so you pay attention to the very end. And if I want to take an extra minute because I didn't finish because you slowed me down, you have to respect me now. I'm almost done though. All right? And then by the time you get to a larger worm, they have, look at this, it's a network of blood vessels surrounding a tube to collect the toxins. This is called a nephron. And then the actual bugs, they have these are worms. The bugs have tubes, like the opposite of the they're called malphagian tubes, that actually excrete the toxins through the tubes and through the skin. So it's kind of like a worm thing. But by the time you get to the segmented worms, you actually have nephrons. Now, fast forward to the time that you get advanced reptiles. Instead of having one nephron, you have a network of thousands of nephrons into something called a meta thing. So our kidney is an enormous network of the nephrons that the worms have. So it's basically a center of nephrons, right? That collects tubes to collect the excretion material. At the end of the year, we return to this topic of the excretion system, but mostly to study how the diffusion that we learn about in this chapter plays a role in the direction in which the tubes travel through the blood vessels in order to collect the waste and repurpose the waste as well. So there's a lot of active transport and diffusion that matters in here. This is a very good example of diffusion that works, and active transport at work. So that's an important one. And then last but not least, so I talk about all of these things. But community ecology now, at the population levels, community level, also saves energy. When a symbiotic life works with another, like the bacteria living in the rumen of cows, right? They digest the cellulose, and then the cows get energy from the cellulose. Bacteria has an endless supply of food. Energy saving for both. That's the whole point of symbiosis, right? To provide for both. Bacteria communities living around these events. One gets one chemical, produces something that the other one needs, that produces something that the other one needs. It's a completely kind of different food web that doesn't work in the same sugar chain that we are used to. But it also works. All about saving energy. Interactions between populations are designed to maximize the use of energy so that we don't waste it. All our systems work together for that purpose, to maximize the use of energy so we don't waste it. That's 4.B.2. Lots that we learn about. Next class, we're going to try to hit 3.A.2 and maybe a little bit of 3.A.3. Review for that. The lab is on Monday. 3.A.2 and 3.A.3 next class. If you have competition, you can do it in your own time. What is 4.A.3?